everyone. Welcome to a new session on Introduction to Bioinformatics. Here we discuss DNA sequencing basic methods. The genetic information of an organism is stored in defining set of chromosomes. The human genome, for example, is a set of genetic information encoded in 46 chromosomes found in the nucleus of each cell. It is the entire set of hereditary instructions for building, running and maintaining an organism and passing life on to the next generation. The chromosomes are organized into 23 pairs. One chromosome of each pair is inherited from the mother and one from the father. The X and Y pair of chromosomes determine the sex of the organism and the other 22 pairs are called autosomes. The human genome is made up of a set of very long DNA molecules, one corresponding to each chromosome. Around 35,000 genes are arrayed along these molecules. In short, the genome is divided into chromosomes. Chromosomes contain genes and genes are made up of DNA. The word genome was coined in about 1930. Even though scientists didn't know then what the genome was made of. Every species of Earth has its own distinctive genome unless you are an identical twin. Though unique, the difference is simply a matter of degree. Genomes are found in cells and it contains the information that affects every aspect of our behavior and physiology. So, study on the human genome is likely to give us insights into different facts of our life. For example, why some people die of heart disease and others die of cancer. Study of genome can be of different ways, like study a very small part of the genome or the genome as a whole. Study the sequence of a gene, the function of a gene and the parts of the genome that regulate genes, location of genes in the genome or investigate how differently genes work together. The genome can be well studied through genome sequencing. Genome sequencing. Sequencing is the first step of structural and functional analysis of newly determined sequence. The importance of sequencing is increasing to find the interface between new protein and the one which already exists. DNA sequencing commonly refers to the methods using to determine the order of the nucleotide bases in DNA. Biological researchers are totally dependent on the knowledge of DNA and are accelerating day by day. Other research areas in which sequencing is making use of are diagnostic, biotechnology, forensic biology and biological systematics. Sequencing started in 1953 by the establishment of DNA double helix by James Watson. The next sequencing was RNA sequencing of first complete gene and genome of bacteriophage MS2 by Walter Fierce in 1972 and 1976 respectively. The large gap between the first two sequencing is because different DNA molecules hold similar chemical properties that made it difficult to separate them. The chain length of naturally occurring DNA molecules was much greater than for proteins that made complete sequencing unapproachable. The existence of only 4 bases in DNA made the sequencing difficult for DNA than for proteins. The DNA sequencing was very difficult without the knowledge of DNases. But after 1970, there were several achievements in DNA sequencing. In 1973, Walter Gilbert and Alan Maxam reported the sequence of 24 base pairs using a method known as wandering spot analysis at Harvard. In 1977, Frederick Sanger developed a sequencing method at the MRC Center, Cambridge. The complete DNA sequence of the Epstein-Barr virus with 170,000 base pairs long was sequenced by the scientists of Medical Research Council in 1984. Then The first semi-automated DNA sequencing machine was developed by Leroy E. Hood's laboratory at the California Institute of Technology in 1986. In 1990, large-scale sequencing trials on Mycoplasma capricolum, Escherichia coli, Caenorhabditis elegans and Saccharomyces cerevisiae has started by US National Institute of Health at a rate of $0.75 per base. The first complete genome of free-living organisms, Haemophilus influenzae, the bacterium, is sequenced by Hamilton Smith and colleagues at the Institute for Genomic Research in 1995. Since 1990s, the usage of sequencing methods has increased day by day. The sequencing methods that are discussed here are 1. The Sanger method and its most important variants. 2. The Maxam-Gilbert method and other chemical methods. 3. The PYROSEQUENCING method, DNA sequencing in real time by the detection of released pyrophosphate. And 4. Single molecule sequencing with exonuclease. Exonuclease digestion of a single molecule composed of a single strand of fluorescently labeled deoxynucleotides. Maxam-Gilbert sequencing. This is a method of DNA sequencing developed by Alan Maxam and Walter Gilbert in 1977. This method is based on nucleobase specific partial chemical modification of DNA and subsequent cleavage of the DNA backbone at sites adjacent to the modified nucleotides. This method is coming under the first generation DNA sequencing. This is a chemical cleavage method using double-stranded DNA samples and involves modification of bases in the DNA followed by chemical base specific cleavage. The base specific cut occurs at a small proportion of either both purines, that is adenine and guanine, or preferentially at A, or the pyrimidine, that is cytosine or thymine, or at cytosine only. The resulting DNA fragments are then denatured, run side by side in slab gel electrophoresis, autoradiographed and analyzed. Purified DNA can be read directly. Homopolymeric DNA runs are sequenced as efficiently as heterogeneous DNA sequences. The method can be used to analyze DNA protein interactions, which is a part of DNA footprinting. Also used to analyze nucleic acid structure and epigenetic modifications to DNA are some of the advantages of this method. But this method is not so commonly used these days because it requires extensive use of hazardous chemicals. It has a relatively complex setup, technical complexity. It is difficult to scale up and cannot be used to analyze more than 500 base pairs at a time. The read length decreases from incomplete cleavage reactions and it is difficult to make Maxam-Gilbert sequencing based DNA kits. Initially, all the steps of these chemical sequencing methods were performed manually. Years later, a system composed of a computer controlled microchemical robot that carries out of one of the 4 reactions, that is guanine, adenine plus cytosine, cytosine plus thymine or cytosine alone, in less than 2 hours was described. The key points in the Maxam-Gilbert methods are the chemical reactions. They can be separated into 2 different groups. 1. 4-lane methods where 4 or more separate cleavage procedures are used and the information is displayed in 4 or more parallel gel lanes, and 1-lane or 2-lane method where all reactions are based on only one chemical modification and electrophoresis is performed in a single or 2 lanes. The first report of a single lane method was based on a chemical cleavage procedure that uses hot aqueous piperidine for several hours. In 1991, Negri et al. Described a 2-lane method which can become 1 lane by mixing the products of the 2 reactions, where the labeled DNA fragments is heated in the presence of formamide. The result is an efficient cleavage of the phosphodiester bond at the 3' residues adenine, cytosine and guanine with the relative efficiency A is equal to G greater than C. The bias between A and G is obtained through pre-treatment with methylene blue. In conclusion, the main advantages of the Maxam-Gilbert when compared with methods are: 1. A fragment can be sequenced from the original DNA fragment instead of from enzymatic copies. 2. No subcloning and no PCR reactions are required. Consequently, for the location of rare bases, the chemical cleavage analysis cannot be replaced by the dideoxynucleotide terminator method, as the latter analyzes the DNA of interest via its complementary sequence. It can thus only give sequence information in terms of the 4 canonical bases. 3. This method is less susceptible to mistakes with regard to sequencing of secondary structures or enzymatic mistakes. Despite of all these advantages, most of the protocols have some drawbacks. First, the chemical reactions of the protocols are slow and the use of hazardous chemicals requires special handling care. The worst problem is the occurrence of incomplete reactions that decreases the read lengths. Sanger's method or chain termination method. The first method described by Sanger and Colson for DNA sequencing was called plus and minus. This method used Escherichia coli DNA polymerase 1 and bacteriophage T4 DNA polymerase with different limiting nucleoside triphosphates. Ionophoresis on acrylamide gels has been used to resolve the products that are generated by the polymerases. Due to the inefficacy of the plus and minus method, Sanger and his co-workers described a new breakthrough method for sequencing oligonucleotides via enzymatic polymerization. This method was initially known as the chain termination method or the dideoxynucleotide method. It consisted of a catalyzed enzymatic reaction that polymerizes the DNA fragments complementary to the template DNA of interest. Briefly, a p32 labeled primer was annealed to a specific known region on the template DNA, which is a starting point for DNA synthesis. Catalytic polymerization of deoxynucleoside triphosphates onto the DNA is occurred with the help of DNA polymerases. The polymerization was extended until the enzyme incorporated with a modified nucleoside known as terminator or dideoxynucleoside triphosphate or ddNTP into the growing chain. This method was performed in 4 different tubes, each containing the appropriate amount of one of the 4 terminators. All the generated fragments had the same 5' end, whereas the 3' end was determined by the dideoxynucleotide used in the reaction. After all the 4 reactions, the mixture of DNA fragments with different size was resolved by electrophoresis on a denaturing polyacrylamide gel in 4 parallel lanes. The pattern of bands showed the distribution of the termination in the synthesized strand of DNA, and the unknown sequence could be read by autoradiography. In this figure, you can see the various steps in Sanger sequencing method. A single stranded DNA template, a DNA primer, a DNA polymerase are involved. To each reaction, only one of the 4 dideoxynucleotides such as ddATP, ddGTP, ddCTP or ddTTP are added. Following rounds of template DNA extension from the bound primer, the resulting DNA fragments are heat denatured and separated by size using gel electrophoresis. The base, that is at the end of each strand, has a fluorescent tag attached, a different color for each basis. These colors can be seen with a fluorescent microscope, and the sequence of the DNA is determined by the sequence of colors that come up on the gel. A computer reads the colors and determines the sequence, which it then combines with all other sequences to make up the whole genome. Now, we can study the different approaches in Sanger sequencing method. 1. Direct approach. In direct approach, the sequencing of unknown DNA is performed within the sites in which the sequence is known. The unknown DNA sequence is amplified by inserting it into a vector. The first sequencing reaction is performed using the primers that hybridize to the vector sequence and polymerize the strand that is complementary to the template. Then, the second priming site has been chosen in the newly generated sequence by following the same direction as the first reaction. This is known as primer walking, and it helps to reduce the redundancy. However, it requires the synthesis of each new primer, which in the past was time consuming and expensive, especially when dye labeled primers were used. Nowadays, the appeal of a cost effective and time saving method that uses small oligonucleotide libraries has disappeared with improvements in primer synthesis technology. However, the demand for a sequencing method that was able to provide long read length, short analysis time, low cost and high accuracy has led to several modifications of the original Sanger method. In addition to several improvements in the procedures and in the reagents used in the sequencing reaction, further development in DNA separation technology was of paramount importance for the completion of the human genome project. The next method is Dye-terminator sequencing. Improvements in the DNA polymerase enzyme increase the quality of the sequencing reactions and data. Isothermal DNA polymerases were used at the beginning for manual and automated sequencing. The reactions were performed at 37 degrees Celsius for 20 minutes. That helps the T4 and T7 polymerases to incorporate with all 4 terminators. But these polymerases are very sensitive to temperature that can easily be deactivated by a temperature fluctuation. With the discovery of the polymerase chain reaction and the use of a heat stable DNA polymerase called Taq polymerase, it was possible to perform sequencing reactions with reduced amounts of DNA template compared to isothermal enzymes. In this approach, dideoxynucleic acids or ddNTPs are used instead of deoxynucleic acids. Although the approach is largely improved, there was still significant variation in peak intensity for fluorescently labeled dye terminators. The pattern of the termination was reproducible and predictable, but this variation made automatic base calling difficult. A few years later, one of the major suppliers of fluorescent sequencing kits introduced a modified set of fluorescent labels for ddNTPs. With this new dye-terminator kit, the signal was more even and automated base calling improved significantly. The next method is automation and sample preparation. Automated DNA sequencing instruments or DNA sequencers can do 24 runs a day with 384 DNA samples in run. Size separation, detection and recording of dye fluorescence are performed using electrophoresis, and data output is getting as fluorescent peak trace chromatograms. A number of commercial and non-commercial software packages can trim low quality DNA traces automatically. These programs score the quality of each peak and remove low quality base peaks, which are generally located at the ends of the sequence. An example for the output file is shown in the figure. In short, Sanger sequencing provides a high degree of accuracy, long read capabilities and the flexibility to support a diverse range of applications in many research areas. Sanger sequencing is highly recognized for DNA sequencing applications, but also supports applications in RNA sequencing and epigenetic analysis. Also in Sanger sequencing, each individual reaction is fairly cheap and very accurate. Random approach or shotgun sequencing. This is random process in which there is no control of the region that is going to be sequenced, also known as shotgun sequencing. Genomic DNA is randomly fragmented using nebulization or sonication into smaller pieces ranging from 2 to 3 kb. The DNA fragments are inserted into a vector that is replicated in a bacterial culture. The DNA fragments are extensively sequenced through several positive amplifications. The generated sequences may overlap because of the random nature of this process. A high level of redundancy of this process makes the approach very expensive. Venter et al. Introduced a new version of this method in 1996 in which the whole genome can be shotgunned at once. Shotgun sequencing is well established because of the availability of the cloning vectors, fluorescently labeled primers, software for base calling and sequence assembly. A simple diagram of shotgun sequencing is shown in the figure. Although this approach is very compatible with automation, the gap produced by this method can be completed only through direct approach. As a conclusion, shotgun sequencing is a much faster process than clone by clone sequencing. Whole genome shotgun sequencing uses a fraction of the DNA that clone by clone sequencing needs. Shotgun sequencing is particularly efficient if there is an existing reference sequence. Also, it is much easier to assemble the genome sequence by aligning it to an existing reference genome and less expensive methods requiring a genetic map. PYROSEQUENCING. This is a simple and easy technique for DNA sequencing by synthesis. It is a real-time DNA sequencing method based on the detection of the pyrophosphate released during the DNA polymerization reaction. Initially, this method was used to monitor the DNA polymerase activity. In the first step, pyrophosphate is released when one of the deoxynucleotides, that is DATP, DCTP, DGTP or DTTP, is incorporated into the chain extension by DNA polymerase. This released pyrophosphate is then converted into ATP by ATP sulfurylase, and the light is emitted via the firefly luciferase that catalyzes luciferin into oxyluciferin. The average number of emitted photons per template chain in a given step is proportional to the number of deoxynucleotides incorporated per chain at that step. The sequence can then be determined by simply noting if incorporations occur and by counting the number of incorporations in a given attempt. The amount of light emitted can be measured by an avalanche photodiode or photomultiplier tube. The main applications of this method includes the analysis of secondary structure such as hairpin structures, analysis of single nucleotide polymorphisms, mutation detection and de novo DNA sequencing for short and medium length DNA. High throughput sequencing. The need for low cost sequencing has turned into the development of high throughput sequencing technologies that parallelize the sequencing process, producing thousands or millions of sequences at the same time. High throughput sequencing technologies aim to lower the cost of DNA sequencing beyond what is possible with standard methods. Now let us study the different high throughput methods. First one, Lynx Therapeutics Massively Parallel Signature Sequencing or MPSS. MPSS was developed in the 1990s at the Lynx Therapeutics, a company founded in 1992 by Sidney Brenner and Sam Elter, which is the first of the next generation sequencing. By applying this method, almost every transcript in the sample will be revealed and provide the expression level with high accuracy. MPSS was a bead-based method that used a complex approach of adapter ligation followed by adapter decoding, reading the sequence in increments of 4 nucleotides. This method made it susceptible to sequence specific bias or loss of specific sequences. This method is typically used for measuring the cDNA expression level. Lynx Therapeutics merged with Solexa in 2004 and this company was later purchased by Illumina. Second one is Polony sequencing. It is an inexpensive multiplex method with high accuracy and is used to read millions of immobilized DNA sequences in parallel. This technique was developed by Dr. George Church in Harvard Medical College. Combination of an in vitro paired tag library with emulsion PCR, an automated microscope and ligation based sequencing chemistry has been used to sequence an E. coli genome. The accuracy of the sequencing was greater than 99.99% with a cost of 1/10 that of Sanger sequencing. The next one is Illumina or Solexa sequencing. This is a sequencing technology developed by Solexa based on Dye-terminators. In this, DNA molecules are first attached to primers on a slide and amplified. This is known as bridge amplification. Unlike PYROSEQUENCING, the DNA can be extended only one nucleotide at a time. The images of the fluorescently labeled nucleotides are taken by a camera, then the dye along with the terminal 3' blocker is chemically removed from the DNA, allowing the next cycle. The next method is solid sequencing. The technology for sequencing used in ABI solid sequencing is oligonucleotide ligation and detection. In this, a pool of all possible oligonucleotides of fixed length is labeled according to the sequenced position. The sequencing provides better results compared to Illumina sequencing. Next method is single molecule sequencing with exonuclease. Single molecule sequencing was initially conceived as a laser based technique that allows the fast sequencing of DNA fragments of 40 kb or more at a rate of 100 to 1000 bases per second. This technique is based on the detection of individual fluorescent nucleotides in a flowing sample stream. The method is divided into the following steps: fluorescent labeling of the bases in a single fragment of DNA, attachment of this labeled DNA fragment onto a microsphere, movement of the supported DNA fragment into a flowing buffer stream, digestion of the labeled DNA with an exonuclease that sequentially cleaves the 3' end nucleotides, and detection and identification of individual fluorescently labeled bases as they cross a focused laser beam. Let us now summarize what we have learned in this session. 4 best known basic DNA sequencing techniques are Sanger sequencing, Maxam-Gilbert method, PYROSEQUENCING method and high throughput sequencing. There are good prospects for the emergence of DNA sequencing method which may revolutionize the field of sequencing. The sequencing methods that are in development are nanopore DNA sequencing, tunneling currents DNA sequencing, sequencing by hybridization, etc. In the end, the choice of sequencing technology can be totally depend on the specific research question. Here are a few assignment questions for you to work out. 1. How different is one human genome from another? 2. How does the sequencing machine know whether a base is an A, C, G or T. 3. How do you assemble a genome? 4. What is the difference between a genome mapping and genome sequencing? 5. What makes sequencing the human genome different from sequencing other genomes? Here I can suggest a few books for your reference. Sequence Evolution, Function, Computational Approaches in Comparative Genomics, written by Kunin E.V. and M. Galperin, published in the year 2013 by Springer US. Genome Sequencing, Technology and Algorithms, written by Kim S., H. Tang and E. Mardis, published in the year 2008 by Artec House. The $1,000 Genome, The Revolution in DNA Sequencing and the New Era of Personalized Medicine, written by Davies K., published by Free Press in the year 2010. Automated DNA Sequencing and Analysis, written by Adams MD, C. Fields and J. C. Venter, published in the year 2012 by Elsevier Science. A Life Decoded, My Genome, My Life, written by Venter J. C., published in the year 2007 by Penguin Publishing Group. Here are some few websites for your reference: www.genome.gov, www.illumina.com, www.ncbi.nlm.nih.gov. Thank you for watching this program. See you in the next session. Until then, bye.